Um, Hello, good afternoon. Thank you for coming. And I will start this session with a talk about eyes. So we are moving our eyes on a daily basis. For example, when we change fixation point and look at the object, when we are reading, if someone is very annoying, or if we are watching someone. So this motion has a similar feature, because it's essentially the rotation of the eye about an axis. And in this work, we are looking on how the fluids in the eye responds to such rotation. So I'm Maria Dvoryashina, and this is a joint work with Rodolfo Repetto and Jennifer Tweedy, financially supported by Offtec. Okay, so this beautiful figure is a cross-section of the human eye. The bulk of the eye contains two main fluids. It's a vitreous humor located at the back of the eye, and it occupies the majority of the volume. And then the front of the eye has aqueous humor, which is a transparent water-like fluid. So in this work, I will focus on aqueous humor. And we're working in the anterior chamber of the eye, which is the region between the iris, the colorful part, and the cornea, the transparent part on the front. Right, so we're looking at how this fluid moves when the eye is rotating. Why this is important? So potentially this flow is known to be the fastest mechanism of aqueous motion in the anterior chamber, meaning that it probably contributes to wall shear stresses on the cornea, which is, again, this transparent tissue, which in turn, large wall shear stresses might lead to cell detachment, which is bad. On the other matter, aqueous is also responsible for mixing in the anterior chamber and nutrient delivery to our avascular tissues, so that steady component of this flow might also contribute to mixing. So this flow has been studied extensively numerically, in particular two physiological eye rotations. So one is saccadic motion, is when you change the fixation point. So this is one saccade. Or periodic saccades is when you read or watch tennis or something. And another one is rapid eye movements that occur during a REM phase of sleep. Right. So as I said, many numerical works have been done. So this is a three-dimensional time-dependent flow, which makes it a bit challenging numerically to expand and investigate a parameter space. And also, some nice features of the flow are lost during numerical simulations, which I'll show you in a minute. So the purpose of this work is to construct a simple mathematical model based on lubrication theory to solve the problem for this flow and do parameter study. Right, so we consider a simple model where the anterior chamber is this region here, sits on a sphere, and the anterior chamber, so is the region where we're solving. The sphere rotates harmonically with amplitude beta and angular frequency omega f, and the anterior chamber is considered to be a thin domain, so it has a small aspect ratio. The thickness over the width is, we call epsilon, and it's a small parameter in our model. So then the fluid is described by Navier-Stokes equation in the moving frame, so we consider, so the fluid velocity here is velocity relative to the motion of the domain. And to use lubrication theory, we scale up these equations. And the essential feature of lubrication theory is that your velocity along the thickness of the domain is epsilon times smaller than other velocities, which after scaling gives us three-dimensional numbers. So we have epsilon, which is the aspect ratio of the domain. We have alpha, which is a Wormsley number, and beta, which is the amplitude of the rotations. Right, so then getting rid of small parameters of order epsilon squared, epsilon beta, and 1 over alpha squared, we come up with this simplified problem. So this problem is forced by these two terms. The one at the top is due to centrifugal force, and the one here is essentially angular acceleration. And the nice feature of this system is that it's linear. So this is already a huge simplification. We simplify it even further by splitting the solution into three harmonics. So we have a steady streaming component here, which is a time-independent component of the flow. There is a dominant frequency component that has largest velocities, and it's double. So the dominant frequency component is important for wall shear stresses, and the steady streaming time-independent component is important for mixing. So in the further results, will mostly focus on mixing and will not talk about wall shear stresses. But the first result is for dominant frequency component. And I'm usually very proud of this movie, but after seeing yesterday beautiful drops with crowns, I probably shouldn't be. Right, so we're looking here, first thing is, so this is if you look at the domain from the front, so the arrows are the velocities and the colors are the velocity magnitude, and this is the depth average velocity over here. So the eye moves left and right, and it follows the ball. So when the little ball is in the center, you look straight, and then the eye follows the ball. Right, so we can see here the formation of two nice circulations, and actually, well, this is quite interesting because numerically it hasn't been observed, particularly because the numerical simulations are done in terms of absolute velocity, and because of the velocity of the domain, this structure is hidden. Right, so the cross-section on the right is the equatorial plane, so it's a half of the equatorial plane. And again, when the little ball is in the center, you look straight, and then the eye moves as the little ball. And here, there is a formation of boundary layers for large alphas. And yeah, so also we can see that the flow is highly three-dimensional, which is quite nice. Right, so it's probably, okay, sorry. So then we have, this is the steady streaming. So here, the cross-section on the left, this is a time-independent component of the flow, which that's why it doesn't move. So this is the vertical plane, and there is like a circulatory motion in the vertical plane. And the one on the right is, again, the view on the domain on the front. And what is nice is flower-like structures. It has like little circulations over here. So this is potentially important for mixing. Now, in terms of mixing, so potentially there is another flow mechanisms in the anterior chamber, which is a thermal flow. So it's like a buoyancy-driven flow, and it occurs on the vertical plane, just like the steady streaming does. So the presence of this nice structure has potential importance in terms of transversal mixing. So not only along the vertical planes, but also in transversal directions. Okay, so we would like to see the dependency on the parameters and the real eye rotations. So this saccadic motion follows sort of a saccadic law, which relates alpha and beta in this way, which is the Bloch curve. And then rapid eye motions are these points, so this will be maximum amplitude REM and average REM. So we will consider this sort of parameter range. Sorry for that. So we plot both steady streaming velocity and thermal flow velocity. So the steady streaming one are the solid lines, and they represent saccadic motion. And then the symbols are REM, again. And the dashed lines are for the thermal flow. And then you have three different colors are for three different thickness of the domain. So blue is the thin one, red normal, and green is the thick. And then, right, so what we can see here is that in all the cases, the saccadic flow, the steady streaming velocity is as important as thermal flow velocity, meaning that both mechanisms contribute to mixing. And what is even more interesting is that for REM, you have, also the magnitudes are quite similar, but the REM happens when the eyelids are closed, meaning that there is no temperature difference in the anterior chamber and there is no thermal flow. So this is the only mechanism that, that contributes to mixing during night. Okay, so to conclude, we developed a model, a simple mathematical model that describes the flow due to the eye rotations. Our solution shows nice three-dimensional flow structure that has not been observed numerically, and steady streaming contributes to mixing. Thank you.
Any questions? known about whether the fluid changes? Does the viscosity change or things like that?
So I don't think so. I think mostly fluid rheology and things in terms of properties change on the back of the eye, but aqueous always has a water-like property, so it won't affect the fluid properties.
Okay.
If that's a question.
Yeah.
Have you examined what would happen if you move your chamber a little bit up or down the eye relative?
Oh, like you mean change the radius of the sphere essentially? No, we haven't. That's a good question. Thank you.
Okay, thank you.
Thank you.
Yes, and this is work I've done with Jeevan Chakrabarti, who was the postdoc working on this project, who is now a faculty member at IIT Kharagpur. Okay, so what I'm going to talk about today is thinking about how plants grow, and I'm going to come over here so I can see what I'm doing. So why do I care about plants? Well, I like to eat food, and that's where most of our food comes from. So that's kind of my motivation is we want to have enough food to eat. Olivier sort of introduced some of this work yesterday in some ways. So here I've got my plant cells. I particularly think about plant roots, but actually everything I talk about today is going to be generally applicable to other types of plant cells as well. But in the plant root, we've got this really nice system here, which is that in this bit here, which is called the division zone, the elongation zone, cells just grow and they don't divide. So they expand massively in length. They grow about 30 times in 6 hours, and they have very minimal radial changes. And that's kind of interesting. So that's what we're going to talk about. And we haven't got time to show the video, so I'm not going to. Okay, so these cells, so you've got your plant cell is different from your animal cells because they are surrounded by a cell wall, a tough cell wall, which includes oriented cellulose microfibers. So it gives it this sort of anisotropic structure. And that cell wall holds in a very high internal turgor pressure, which on the time scale of growth is constant. So your turgor pressure in your little tiny plant cells is about 3 bar, so it's the same as a car tyre. So they're really high pressure, and this cell wall is holding that in, but allows this massive growth. This turgor pressure stretches, exerts tension in the cell wall. If that tension is big enough, the whole thing will grow, and it has this growth in length, but hardly any growth in radius, despite being an isotropic force because it's a pressure. And how the cell controls its growth is it changes its mechanical properties, so for example the cell wall viscosity, in response to what's going on, and that makes the whole thing grow. So people have used models of growth for a long time, tend to be variations on this Lockhart model from 1965. So this says that the relative elongation rate, so here L is my length and T is time, so how quickly my cell grows is proportional to how much my tension is my pressure is above my yield, with some constant of proportionality, which is my extensibility. And cells can also twist due to the presence of these fibers, so they're going to elongate and they're going to twist. What I'm trying to understand is how all of that works. So I'm just going to think at a single cell level. So I've got my single cell, so I've got thin walls, thin cell wall being inflated by my turgor pressure, which is constant. I'm going to neglect the yield stress. Oliver and I have done some other work looking at where this yield stress comes from, so I'm going to neglect that at the moment. I'm just going to consider a purely viscous material. But I really want to include these reorienting cellulose microfibers, this anisotropy in the cell wall. So in order to do that, I'm going to use this stress tensor. So this is a stress tensor from Ericsson. So if that would be my normal Newtonian bits, so I've got my stress is proportional, we've got pressure, strain rate, and I have all these extra bits which are dependent on my A, which tells me the direction my fibers are looking in. So here I have a kind of an active bit, which is like if I've got a tension in my fibers. I've got, this is my sort of extensional bit. So this says that it's more difficult to stretch my material in my fiber direction. And then this bit is all about the shear stresses. So my shear stress is bigger in my fiber direction than perpendicular to it. And then that director field A also evolves. So my fibers are convected by, stretched by, and reoriented by the motion through my cell wall. So these fibers are going to move, and they're going to affect the mechanical properties. So One of the really nice things about applied maths, I think, is that you can draw analogies between different areas. So I hear thin walls inflated by constant turgor pressure, and because of who my PhD supervisors were, I think glass blowing and Peter Howell and John Ockenden and people like that. So I can take their models and I can modify them using all this stuff, and off I go. So I'm not going to go through all the maths of how you do these things because there's not time. So I've got a thin cell wall, and that means I can do things. If I integrate across the thickness, I can work out sort of average quantities. I'm going to denote that by bar when I come to it. I'm going to think about things in terms of arc length, so along my fluid sheet, and then azimuthal and normal coordinates. So I've got S, theta, and N being my coordinates. My fibres I'm going to assume lie in the plane of the wall, so there's no N component. In that case, I can just write it in terms of an angle phi. So I've got my my fibres, and that can depend on where I am through the cell wall, because as I stretch the whole thing, my fibres are going to reorient. I've got an internal pressure P, which is going to produce my axial tension, and I'm going to potentially impose an external torque, capital sigma, which is going to tell me, like, maybe there are other cells influencing what's going on, or some kind of thing from the environment. I'm going to choose my mechanical properties and my deposition rate of new material to enforce. I'm going to have a constant uniform radius of my cylinder, and I'm going to have a constant uniform thickness of my cell wall. And that kind of works quite nicely. If I'm depositing new cell wall material, though, I need to be depositing new fibres. And so there's an interesting question about when you're depositing new fibres, what angle does that happen at? And I'm going to talk about that in a minute. So, if I start, what I'm really interested in here is growth and the twist. So if I start off with my axial and azimuthal momentum equations, and I integrate them, and I fiddle around with them a little bit and do a bit of algebra, I could end up with expressions for US here, which is my axial velocity, and U theta, which is my azimuthal velocity. And I've written it all in this format because it's an awful lot. You can kind of unpack what's going on by doing it this way. So here my Ks are all my different sort of averaged directional viscosities. Viscosities in inverted commas because they're not technically viscosities because of viscosity times a thickness. It's sort of more of a, because I've integrated across the thickness. So for example, this one here says, okay, what's my extensional viscosity in my axial direction? And you can have the same sorts of analogies for these other two. And then I've got the tension here. So this is my axial tension reduced by any active behaviour I've got in the fibres that are pointing in the right direction. And my torque, again, reduced by any active behaviour I've got from my fibres. And so you can kind of end up with this. So that's interesting because you can show under appropriate conditions that the right-hand sides of both of these are independent of S, which means you can just integrate them and they're going to be linear, so the velocity is going to be linear in S. So my growth of my cell, or my axial velocity at the end, if I keep the bottom of my cylinder fixed, the axial velocity at the end tells me how quickly I'm growing, and the azimuthal velocity at the end tells me how quickly I'm twisting. So you can work out what those are. So here's my relative elongation rate. So it's some balance between the tension and the twisting. And then this is a very common thing to talk about in the plant sciences literature. And then I've made up another thing, which is the relative twist rate, which is telling me how quickly I'm rotating. So I've got my R0 there just to show that it's a rotational velocity. R0 is 1 in my non-dimensionalization. So you can see here I've got some kind of balance between axial tension and imposed torque, which give me some, so how they combine tells me something about how quickly I'm going to grow and how quickly I'm going to twist. So of course I have to, this is all dependent on my angle, and this is the angle equation I get left over, which sort of looks quite nice, and then you remember that these depend on integrals over the thickness of trig functions of the angle. So that's far worse than it looks. sort of, you can express it this way, and then you can kind of unpack what's going on from the physics point of view, but solving it is actually quite difficult. This is interesting because you might expect that as you're stretching your cell wall, your fibres are always going to line up so that they're pointing entirely vertically. That's not always true if you've got a torque. You can actually, if you've got a twist coming in, then it can actually go right the way over and end up going around to the other side. So you get some non-intuitive behaviour going on. Okay, so you can do all sorts of different bits and pieces, and I haven't got time to talk about most of them. So you could do twist. You can let it twist with a constant sigma. You can impose the right sort of sigma to make sure that it doesn't twist at all. You can say, okay, well, what if I continually deposit fibres at a constant angle? Or there's this nice idea in the literature that it all depends on the principal stress. I can whack that in and work out what those have to be. If you choose your sigma so that it doesn't allow twisting at all, then you can get a nice sort of closed form of your Lockhart equation. If you do it for constant angle, in fact, by using characteristics, you can work out a really nice closed form of a sort of Lockhart type equation, which gives you some confidence in your numerics that I'm going to show you very, very briefly. So here I've got, this is for no twist. Here I've got constant angle deposition. Here I've got principal stress dependent angle deposition. And you can see if I've got principal stress dependent, my length sort of starts to saturate rather more than my time did. So in summary, I have very quickly talked about a framework where you can think about these variations across the cell wall and variations in your deposition mechanism, and we've sort of shown how all these different kind of bits compete to tell you about growth and about twisting. Thank you.
Do all the fibres align with one another, or do they ever overlap?
So in this framework, everything is perfectly aligned locally, so that you can get variations through the thickness, but at any given, because it's a continuum model, so at any given point it will be perfectly aligned.
Thanks very much. Yes, so I'll be talking about transport in microvascular networks and in the placenta specifically. So what's very interesting in the placenta is that you have two blood supplies. There's a maternal and a fetal blood supply. And the fetal blood supply is all hosted in these trees. And at the end of these trees are the primary exchange unit of solute, where all the CO2 and oxygen exchange happens. And these exchange units called terminal villi. They're about 200 microns across. And they consist of this fetal vasculature, where the blood goes from and to the fetus. Outside of that is villus tissue. Outside of this blue surface is maternal blood, and between the blue and yellow surfaces is tissue where transport happens through diffusion and active processes. So I'll be talking about an image-based model computational study of solute transport in these terminal villi, which are the primary exchange units in the placenta, and I'll be addressing questions like what limits solute transport, and is it flow, is it geometry, and what's the relationship between the geometric structure of these terminal villi and their physical function. So yes, the model. This is a 3D print of a fetal network. You can have a look at it later. You look at it, it's pretty clear. It's complicated. I mean, the topology is complicated. The segments of this network are not exactly perfect tubes. There's all sorts of bulges and twists. So the way I propose to solve this is to boil down this complexity in three parameters, which are called geometric determinants of transport. These three parameters are they can be obtained with standard computational tools. First one is the resistance. You solve Stokes flow over this kind of capillary domain that gives you the network resistance. The second one is the exchange capacity. You get that by solving diffusion over the villus tissue that surrounds this capillary network. And it's called Nmax, exchange capacity, and it's proportional to a length curly L I'll refer to later. Third one is the capillary length, which you can get by standard imaging techniques, like skeletonization. So once you have these three, I'll also require two dimensionless parameters. The first one is the Damköhler number, which is a ratio of fluxes. It's a ratio of diffusion limited and flow limited flux. So these describe extreme transport regimes, if you like. In the diffusion limited regime, the network operates near its optimum, and in that case, it's the geometry that limits how good transport is. So it's how the capillaries are arranged with respect to the villous surface. In the flow limited regime, the flow is quite slow, and transport is limited by the resistance primarily. And the Damköhler number describes the transition between these two. A second non-dimensional parameter is diffusive capacity. It's purely geometric parameter. Basically tells you how good the network is, can be for diffusion. And it's a ratio of lengths where this length comes, I mentioned before, comes from solving the diffusion equation. And LC comes from measuring capillary lengths. So just to sum this up, I have two non-dimensional parameters, Damköhler number and diffusive capacity, and three geometric determinants, I call them, which you can get from standard computational techniques. So what can we do with this? These are four curves for four terminal villi image-based geometries. What I'm plotting is n on the vertical axis is the amount of material that gets into the capillary. So it's the solute transport into the fetal capillary system. And on the horizontal axis is the pressure drop between inlet and outlet. You can see that basically these look very, very heterogeneous. They look very different, and that kind of translates into this transport scaling. But have a look at what happens when you rewrite the problem in the right variables, in the more natural variables, using the Damköhler number and solute transport scaled by its transport capacity. We get this very nice collapse for all of these very different looking networks. So we've developed a relatively simple algebraic expression that describes the transition from the flow limited regime to the left to the diffusion limited regime to the right. We call it regression equation. And yes, what I want to really kind of get across is that there seems to be this generic scaling, very nice collapse for very different networks. So another thing we can address with this is the question, what is the transport like in real fetal-placental capillary networks? So is it diffusion limited, is it flow limited, or what is it? There's a question that's been asked in literature before, but I think we're the first to have the data and the mathematical framework to answer it because of advances in imaging. And so this is a diagram with diffusive capacity, which describes how good is the network, how good can the network be for transport on the horizontal axis, and vertical is Damköhler number, which you can think of in this case as flow. I've mentioned before, so we've discussed flow limited and diffusion limited regimes, and there's another one that pops up, which is we call weakly flow limited, where concentration boundary layers become strong. So where does the real data lie? For oxygen, for transport of oxygen, it's midway between diffusion limited and flow limited regime, and safely outside weakly flow limited, which means that there's no concentration boundary layers. But we can also discuss with this framework passive transport of other solutes. So we've identified carbon monoxide and fructose, for example, to be strongly diffusion limited, and anesthetic gases to be strongly flow limited. Okay, so everything I've said so far is about transport at the level of a network of a terminal villus. I've in fact compared different terminal villi. But what we can also ask is, what does the transport scaling look like when you zoom in on a network and when you look at individual capillaries like this? So this is a plot of solute transport for 9 individual capillaries, N and J, scaled by their respective transport capacity, plotted over inlet-outlet pressure drop just as before. And you can see that the scaling is different. And one big difference is that it's non-monotonic. In fact, you can have strange-looking things potentially, like a dip below 0 for certain capillaries, and the dip above the transport capillary, capacity for a capillary. And we can explain this with a donor-recipient mechanism. So what's happening is that these capillaries here, for example, the magenta one, act as a donor, and this blue one acts as a recipient, which is due to a flow-counterflow effect. So what happens is that the flow goes through the blue capillary via yellow, magenta, and green, and because of the proximity and opposite direction of flow, blue essentially steals oxygen from its neighbors. So this is what it looks like plotted in the natural variables with a Damm-Köhler number. We have once again a collapse, but there are differences. These truncated capillaries are truncated because they go below zero. It's a log-log plot, so they are our donors. And this isolated top blue capillary is a recipient. So just to make this point very clear, we have non-monotonic scaling at the level of individual capillaries, but we always have monotonic scaling of the transport at the level of a network. And that's because the donor-recipient effect cancels out. So at the level of individual capillaries, some capillaries give and others take, but it doesn't affect the network. So just to sum it up, I've presented an image-based model for fetoplacental solute transfer, and I've discussed how that applies to oxygen and passive transport of other solutes. And I've shown a universal scaling that applies both at the level of a network and at the level of individual capillaries, where we have some exceptions, which we can explain via donor-recipient mechanism and counterflow. So if I were to leave you with one kind of idea, one thought, is that we're showing a generic scaling across terminal villi, despite the quite considerable geometric variability, which you can see just by looking at these pictures of yellow capillaries and blue villus surfaces. That's it. So these are my collaborators, and if you would like to read more, we have a preprint out on arXiv, or you can get it from my website. Thank you very much.
So I was interested to note that this reduction of all these different complex images, you're able to get these sort of universal laws. Can you comment a little bit on what the implications are for sort of as mathematicians doing modelling, if you're wanting to develop these sort of multi-scale modelling laws, models, can these generic scaling be useful for that?
Yeah, possibly. Obviously, we need more data to really confirm this, but if this sort of remains the trend, then we can use essentially an expression for transport that is as simple as this to potentially encapsulate transport at the level of terminal villi, and that would be quite nice to use in a multi-scale model which goes higher. Yeah.
Any other questions? Yes.
What condition do you impose on the blue surface? I don't remember the name. Diffusion.
Yeah. So I'm right. So the transport is essentially an advection diffusion platform equation. So I'm saying that at the villus tissue where the maternal surface is in contact, I have the maternal blood is perfectly oxygenated. Sorry, and I can be more precise. Here we go. So on the blue surface, the concentration is at its maximum, and it comes deoxygenated at the inlet, goes through the capillary system, picks up oxygen, and comes out a little bit more oxygenated, so to speak, through the outlet.
I'm going to have to cut you off.
Sorry.
And follow on and copy.
Thank you.
Hello, I'm a PhD student at the University of Oxford, and my project is a multifaceted collaboration between the mathematics department, where I'm supervised by Sarah Waters and Derek Moulton, and the Nuffield Department of Surgical Sciences, where I'm supervised by Ben Turney, and also an industrial sponsor, Boston Scientific, who are a medical manufacturing company. In particular, Boston Scientific manufacture a medical device called the urethroscope, that's involved in a surgical procedure called ureteroscopy that's used to remove kidney stones. So what the ureteroscope is it's essentially a long, thin pipe that's inserted into the urethra of the patient. It goes through the bladder, up the ureter, and into the kidney. And within this pipe, there's a long, thin, hollow channel that runs that's called the working channel. And also in the wall of the scope, there's a camera and a light that are used to project an image that the urologist then uses to guide their way through the urinary system to find and locate kidney stones. Now, importantly, in order for this procedure to be possible, there's fluid constantly flowing through that working channel. And this fluid serves two primary purposes. Firstly, it washes away any blood and debris, and it also opens up the ureter to allow easier access for the scope. So basically, both of these purposes serve to have better visualization for the surgeon. And the way that the fluid is introduced into the working channel is it comes from a bag of saline solution that's hung at a certain height that causes a hydrostatic inlet pressure that drives the fluid to flow through the scope and into the kidney. As well as fluid flowing through this working channel, working tools are also introduced into this same channel that the fluid flows. And these working tools are either laser fibers or stone baskets, and they're used to obliterate the stone into lots of little fragments or to capture pieces of the stone and to bring them out. Now, the problem is, when these working tools are introduced into the same channel that fluid is flowing, they pose a resistance to the flow, and hence we have a loss of irrigation, and subsequently they can cause a loss of visibility. So this is a challenge. So we addressed this by posing two mathematical modeling questions. The first one is we wondered whether we can optimize the irrigation flow through this channel by modifying the cross-sectional geometry. And we thought about modifying the cross-sectional geometry in two different ways. Firstly, we looked at moving the position of the tool within the channel, and secondly, we looked at changing the cross-sectional shape of the channel. And then the second modeling question I'll talk about is we wondered where would a tool naturally sit within this channel if there's a fluid axially flowing. So to develop a mathematical model to describe fluid flow through the working channel, we have some assumptions. We firstly assume that this channel is long and thin. That seems like a valid assumption. In actuality, it has a radius of about 0.06 centimeters, and it's around 79 centimeters in length. We also assume that the tool and the channel remain coaxial, and that they have uniform cross-sections. Then, under these assumptions, our Navier-Stokes equations reduce to a single equation for unidirectional fully developed flow down the scope, driven by this axial pressure gradient. And we can now look at how changing the position of the tool within the channel will affect the flow. So here we're looking at some cross-sectional velocity profiles where this outer circle is the working channel and this inner black region is the cross-section of the working tool. Now, if we firstly solve with the tool in the center of the channel, we can compute the flow rate, and here I've shown moving the tool from the center to the edge. And we can see by looking at these velocity color maps and the corresponding flow rate numbers above them that the flow rate increases quite significantly as we move the tool from the center to the edge of the channel. And also that the position at the edge of the channel is the one that minimizes the axial fluid resistance. So now that we've thought about moving the position of the tool and how that affects flow, we looked at changing the cross-sectional geometry of the channel. And in particular, we looked at elliptical shapes, and we looked at elliptical shapes for two reasons. Firstly, because they're relatively simple for Boston Scientific to manufacture, and also because the ureter itself is actually elliptical in shape, so we thought that this would be relatively easy to hopefully pass up into the patient. So here, this plot is showing the volumetric flow rate in the y-axis, and on the x-axis, we have the eccentricity value of the outer elliptical channel. And for each of these shapes, we've constrained the cross-sectional area available for fluid flow. And you can see that there's actually an optimal eccentricity value for the channel that gives us the maximum flow rate. And that optimal eccentricity corresponds to the value where the tool can still sit at the furthest edge of the channel. And moving from having circular cross-sections with the tool in the optimal position to having an elliptical cross-section actually gives us a 50% flow increase, which led to some potential for design modifications for Boston Scientific. However, this design modification is requiring that the tool is sitting at the edge of the channel. And in actuality, we don't know where the tool is sitting. So we wanted to look at that question, and specifically, will the tool position itself naturally so that the flow resistance is minimized? So now we're looking at a different resistance question. At first, I was talking about resistance to the axial flow down the channel, and now I'm thinking about resistance to movements of the tool within the cross-section. We can calculate the forces imposed on the outer boundary of the tool by the fluid by looking at the viscous stress tensor. And if we just have a stationary tool, then the force is going to be 0 because we don't have any cross-sectional pressure gradients. So the way that we posed this problem is we thought about perturbing its initial position. And here in this inset diagram, I've labeled the position of the center of the working tool as capital X, and its rotational position, theta, which is the angle made by the major axis of the tool with the x-axis of the domain, and we prescribe these initial velocities, a translational velocity dx dt, and a rotational velocity d theta dt. So we pose the problem in this manner, and we look at an asymptotic expansion, where here our small parameter epsilon is the aspect ratio of the scope, so the ratio of its radius to its length. This problem decouples. And at leading order, we just recover unidirectional axial flow that we've already considered. And then at first order, we actually have Stokes flow in the cross-section that are driven by these prescribed velocity gradients. So we can then solve Stokes flow and get the velocity profiles within the cross-section. And from these, we can compute the forces and torques imposed on the surface of the tool by the fluid. And then we can plug these forces and torques into Newton's equations of motion to see how those forces would drive the motion of the tool within the cross-section. So because this problem is linear, for a given cross-sectional geometry, we can solve the problem once for a velocity in the x direction, velocity in a y direction, and a rotational velocity, and then any movement within the domain would just be a sum of those. And we recover a resistance matrix. And I'm just going to focus on these four coefficients in the top left, where kxx corresponds to the opposing force by the fluid in the x direction when you try and move the tool in the x direction. Kxy is the force in the y direction when you move it in the x direction, and so on. And if we compute the eigenvectors and the eigenvalues of this 2x2 matrix, those correspond to the two orthogonal directions where it's easiest to move the tool and hardest to move the tool, and the eigenvalues are their corresponding magnitudes of those resistances. So we come up with a metric for a most stable position, which is when we try and move the tool in that direction that it's easiest to move it, it will have a maximum opposing force. And we actually see that the position that's most stable is also this same position that gave us the highest axial flow rate. So now that we have these velocity profiles and we have this resistance matrix, we can solve for trajectories. So we prescribe an initial position and rotational angle for the tool, and we give it some initial velocities, a translational velocity and perhaps a rotational velocity. From that, we can calculate the opposing forces and torques, use Newton's equations to then update the velocities and subsequently update the positions, and it's an iterative cycle. So here are two example trajectories, where in both cases we've moved the tool in this direction of minimum resistance. And we can see that as time increases, the axial flow rate down the length of the working channel increases as well. So in summary, these working tools provide a resistance to the flow which can be detrimental because it causes poor visibility to this procedure. We found that the axial flow rate will be minimized by having the tool at the edge of the channel and also by having an elliptically shaped channel. And we also found that the position which gives us the maximum axial flow rate is also the most stable, which indicates that perhaps when Boston Scientific makes these design modifications, they won't have to constrain the tool to sit in the optimal position, and it will sit there naturally. Thank you very much. I'm happy to answer any questions.
Why can't you just increase the pressure of the fluid coming in?
That's a great question. So one of the main challenges is that they don't want to cause really high pressures within the kidney because this fluid is going directly into the kidney. So really, the optimal design would be to have a large flow rate so that they can see what's going on, but not to cause high pressures within the kidney.
Can the instrument not damage the edge of the channel if you keep pushing it to the edge?
So this the channel itself isn't the actual ureter. This is just a scope that's passed up. So if you're moving the tool to the edge of the channel, it shouldn't actually damage that, as it's a rigid material.
My question goes in the same direction, kind of.
Yeah.
If you have done this two-dimensional, right, just in cross-sectional, would you not expect in certain conditions you can have oscillations forming in terms of you can travel down the channel?
Yes, exactly. That's a great question because the ureter itself is not a straight channel. Obviously, it's going to be curved. And we actually think that that will then cause some axial offset, which will naturally cause these cross-sectional pressure gradients. And so then you wouldn't have to prescribe those initial perturbations. So thank you. Yeah, exactly. So from what I've heard from my urological supervisor, I don't think that happens. I think it's a material that manages to stay straight through the channel. Thank you very much.
Hi there, thanks for coming. I'm a first year PhD student at University of Bath, and today I'll be talking about predicting transitions to turbulence in Newtonian and shear thinning blood. So I'm going to start off with a scary statistic. Cardiovascular diseases cause quarter-of-all deaths in the UK. However, we can overcome these through the use of medical devices such as stents for narrowing of vessels, prosthetic heart valves for valve stenosis, and ventricular devices for end-stage heart failure. However, when we introduce these devices into the body, they can cause changes to the hemodynamics, and this in turn can affect both the device and the blood cells. Complications mainly arise due to the fact that blood is multi-phase fluid. A lot of modelling and experiments don't assume this, and in blood we have red blood cells, white blood cells, platelets, and other proteins within it. And when we look at the viscosity, we can see, when we look at the viscosity, we can see that it has shear thinning behaviour. So if we look at low shear rates, aggregability of the red blood cells cause a higher viscosity. Where we look at higher shear rates, the formability of red blood cells cause lower viscosity. And another thing we don't think about with blood is turbulence. So it exists when we have devices implanted into the body, and when we have diseases such as stenosis, where we get this flow separation and a lot of coherent structures develop downstream of the vessel. And this is only a recent type of investigation, and most recent experiments have looked at how blood has a different critical Reynolds number compared to a Newtonian blood analogue. So when we're looking for a vessel, blood transitions at a different Reynolds number compared to analogue of water glycerol or something like that, which is commonly used in experiments. In addition to this, shear thinning fluids have shown to have differences in transition to turbulence. So using these two ideas, the aim of this project is to compare the difference in velocity fields of Newtonian and shear thinning blood in transition to turbulence. So kind of go off topic a bit here. I'm going to look at backward facing step. You most probably don't think of it as a biological geometry, but it can be used to explore mechanical heart valves due to flow separation, in addition to blood damage, where we look at thrombus formation and other blood damage. So first investigations were conducted by Armale, and he said with increasing Reynolds number we get a variety of recirculation zone lengths developing downstream of the step with varying lengths. So we solve the filtered Navier-Stokes equation. We use it at LES, and we use a finite volume solver open foam, and we have a 3D domain, and we discretize with hexahedral cells using multi-blocks meshing method. We apply the following boundary conditions to the domain. We have cyclic boundary conditions to ensure three-dimensionality, and we use second-order temporal and spatial discretization scheme. We use the Pisa algorithm for pressure-velocity coupling for better convergence and due to transient nature of simulation. So our models are looking at two viscosity models. So the first one assumes a constant viscosity. So the Newtonian has a constant viscosity of 0.0035 pascals second. And as you can see, it's a straight line. And when we want to model the shear thinning, we use a Carreau viscosity model, which has this curve. And as you can see from the red circles blood, and the dashed line is the Caro model, which fits quite well. We simulate flow from Reynolds number of 50 to Reynolds number of 7,000. We're most likely not to get that high Reynolds number. It doesn't seem high for this, but it's high for biological flows. So up to 7,000, and it's based on the inlet, 2 times the inlet height. So we have quite a few LES simulations to perform. So we use HPC at University of Bath, Balena, and National Tier 2 Service, Cirrus. So on to results. So as said earlier, the main result you obtain from backward phase step is recirculation zone length. So if I just quickly go back, so this recirculation zone length is the one we're mainly looking at. And you can see up to Reynolds number 400, we have good fit. However, when we increase Reynolds number up to 1,200, we have a less of a fit with the experimental data. This is mainly due to time convergence, where if we just run them longer, the recirculation zone length will reach the experimental, so that's ongoing. We can also compare the Newtonian and shear thinning blood models. So if we look at recirculation zone length for the shear thinning model, this one, it's much lower than Newtonian up here. We can compare this with past experiments where they look at wall shear stress. And if we look, so that was up to Reynolds number of 400. If we look up to 1,200, we see actually the recirculation zone length for both models actually reach the same length at 1,200. And yeah, as you can see here, we have very similar recirculation zone lengths. So to assess transitions turbulence, we take we monitor the velocity downstream of the step, and we monitor the instantaneous velocity fluctuations. So at around the number of 800 and 1,000, we have little to no instabilities, but as we reach 1,200 to 2,000, we can see small fluctuations. And I think the most obvious thing is when we look at the Reynolds number of 2,000, we can see this intermittent fluctuation in the Newtonian model compared to the shear thinning model. And we can also look at the Reynolds number up to 7,000. We can see at 3,000 they actually both have transitional fluctuation flow, and then up to 7,000 fully turbulent fluctuations. So we can look at the turbulent kinetic energy spectra, and for the Reynolds number 1,200, we get fairly similar energy spectra. But when we look at a Reynolds number of 2,000, we get much more difference, much bigger difference between the two models. And so we could, oh, here's a video. So this, the top one, this is at Reynolds number 2,000, and the top one is Newtonian blood, and bottom one is shear thinning. And we can see there's a bit of difference in velocity fields for each model. So we can say transition occurs between Reynolds number of 2,000 and 3,000. It's quite hard to get an exact model, which I try solving more models at the moment. However, this new areas come into play, which looks at the Reynolds number in particular. The Reynolds number we used was based on a high shear rate viscosity. However, for the shear thinning model, the viscosity is changing across the domain. So if we spatially average the viscosity across the domain, the Reynolds number actually changes, which actually reduces the Reynolds numbers. So for instance, at a Reynolds number 1,200 expecting, we're actually getting a Reynolds number 1,000. So when more models are developed, it might actually show that there's no difference in transitions turbulence. There might not be, there might be the same critical Reynolds number, which is an ongoing investigation. So This, I just want to say, is ongoing work. We could say shear thinning is possibly delayed, but it ultimately depends on the Reynolds number and our definition of viscosity. In addition to shear thinning, viscoelasticity of red blood cells might have an impact. So the multi-phase behaviour of blood might explain transition to turbulent better. Future work will be developing more models, LES models, and measuring turbulent parameters of whole blood, so we can compare our models using Doppler ultrasound and PIV. And the final stage will be develop a multi-phase model of blood. I just acknowledge EPSRC for funding, University of Bath for HPC time, and CIRRUS for HPC time. And thank you.
Questions? Yes.
What Reynolds numbers do you expect in the extreme case?
So for well, this is the first time doing a backward phase step because the Reynolds number is based on a square inlet. But for a circular pipe vessel, transition should occur for blood at about 1,800. So, but it depends on the geometry of the.
In the body, if you have a medical device?
I can't say exactly because it would depend on the device, but it could vary.
So, what is the characteristic length of the scale of the flows you are considering?
Well, for our, I don't really know, but if we consider blood, the Kolmogorov scale can reach the size of red blood cells. So this could be 7 micrometers, things like that. Thank you.
I think a slight detour from the current topic of healthcare. So I'm from the University of Leeds. I'm going to talk to you about my work, which is looking at the development of an Eulerian Lagrangian agent method model, which is used to predict the response of fish to hydrodynamic cues in rivers. So a small amount of context first. We have a lot of rivers in this country and around the world, and almost all of them are in some way anthropogenically interrupted. So that's us doing things to the river for whatever reason, be it facilitating navigation or permitting extraction or any of those kind of things. So historically, the ecological impact of this wasn't really assessed, it wasn't really looked at, but it's been revisited reasonably recently, now that we know. Obviously, it affects the longitudinal continuity of the river, so basically migratory fish can't migrate up things like this. So the best option would be to just remove these structures altogether, but they perform quite necessary functions, so it's not really practical. So instead, we look at mitigation through the installation of things like fish passes. So there are hundreds of different designs of these kind of things. They are often only designed for a specific species and neglect all the other species, really. So this leads to no real agreement between what is good and what is not. And similarly, or furthermore, when a new design is appreciated, the only real way of understanding the efficiency of the passage solution is through physical testing. And physical testing involves, the capture of fish, the placing them at the bottom of set designs. And it's expensive, it's invasive, and it's very impractical for large scales of experiments because learned behavior of fish, etc., means that you can't actually use the fish multiple times. So how do we plan on overcoming this? So the idea is, the project aim is to develop some way of computationally assessing the efficiency of fish passes. And this is done by a combination of CFD along with agent-based modeling, which will allow us to basically reduce the need for physical experimentation. So it requires a number of things. So we need some form of quantification of the environment of the river system, which is what's provided by the CFD. So that's environmental data in terms of bathymetry and velocity fields, pressure fields. We also need some understanding of how the fish actually interacts with its environment. How does it perceive its environment, and to what level can it perceive the environment? And then we need some kind of decision-making framework which will allow us to decide how the fish actually reacts to a given stimulus. So the CFD, this is the only slide on CFD, and basically all we are doing is taking some example data set. It's for the code simplification, it needs to be three-dimensional uniform, no temporal dependency, and then the only things that we really need are those, so velocity field, pressure field, and the bathymetry. And it's also worth noting that in the model, the fish has no effect on the fluid, but the fluid does have an effect on the fish, so it's one-way coupled. The reason that we did this, we did explore the idea of two-way coupling, but the complexity is quite large. And also, if you actually sum up the amount of energy expended by a single fish within a river and compare it to the actual energy of the river itself, it's negligible, so it's really not worth it. So once we have our environment, then we need to understand how does the fish react to its environment, how does it perceive its environment. So fish navigate using a combination of different mechanosensory systems. The first two, visual and oral, that's the same as us, but they don't really use those to assess hydrodynamics, so I won't talk about those. And then the swim bladder is an interesting organ that passively changes its volume based on local hydrostatic pressure, so we can really just think of it as a depth sensor. And then they also have the super interesting lateral line system, which is this squiggle along the side of the fish. And that's a series of depth sensors, or pressure sensors, sorry. And that series of pressure sensors will allow the fish to infer pressure gradients along the side of its body. And from inferring pressure gradients, it can infer the motion of the fluid. So really, it's the same way that you could feel wind on your face, except for the fact that theirs is much more sophisticated, so they can feel the motion of the fluid at a distance from their own body. So from here, we can kind of formulate, okay, so there's some volume around the fish where the fish knows what's going on. So we can assume that each fish within the model can access the environmental data from the output of the CFD, and it can look up, basically, the data around itself within a certain local environment. So once we have that, we can move on to some agent-based model. And the agent-based model requires two things: an environment, which we've covered, and the agents themselves. So agents are autonomous, they aren't controlled, they're self-contained, and they have a number of different internal properties. Some internal properties stay the same, such as physical size, and some of them need to be able to vary. So things like hunger levels may increase, and then they also have these internal decision-making processes called methods, and methods are basically reactions to different stimuli. So if you are hungry, one of the methods will be, okay, hunger over a threshold, let's do something about it, let's go and find food. So once we have all those things, those three things, we can start building up the model framework. So we start with the Eulerian mesh, nice and simple, output from our CFD, and then we add a fish into the domain, and we give the fish some physical boundary. So we ensure that whatever the fish does it physically appropriately and doesn't try and fit through tiny gaps, which it used to. And then we add in a Lagrangian reference frame, which gives us our sensory ovoid. So within this ovoid, this three-dimensional volume, the fish is able to sample wherever it wants because it knows what's going on within there. So it samples in there. So once we've got to this stage, we can start talking about the time stepping. So each fish time step, each fish will then sample its local area, and it will make a decision, one single movement decision, based on what it's perceived around itself. So I don't really have time to talk about the actual rule set, but examples are things like rheotaxis. So rheotaxis is a observed behavior in fish where they tend to orientate their nose upstream, and this is because of this is where the data comes from. It's where prey comes from, but it's also where data comes from. They rely a lot on their sense of smell. So if they point their nose upstream, it's a lot easier to smell what's coming. So that's very simple when you have this kind of model, because it can easily sample the area, figure out the average velocity, figure out which way is upstream, and sample it and orientate itself successfully. So other example rules, these kind of things. So this is what I'm doing at the moment. It's basically trial and error of different rules to try to figure out how to get these fish to act in a realistic manner. So there's a lot of literature on what fish like and what they don't like in terms of hydrodynamics. So there's quite a few papers that talk about they don't like certain thresholds of turbulent kinetic energy, and they really don't like high shear, those kind of things. So these are the things that I am including into the model as I go along. So I'll leave you with a nice picture. So this is my example domain. It's about 13 meters long, 6.5 meters wide, 50 centimeters deep. It's a stretch of rivers somewhere in England, but I can't remember where. And that red line is my predicted fish having a nice little swim through. So thank you very much. Happy to take any questions. For the fish, it's simplistic. They're spawned at a user-defined location. So the whole software is being built so that practitioners can use it, basically. If they want to include a fish path in a river, then they can use this tool to see if their fish path will work. So it's a user-defined location of where the fish are created. And then the only boundary conditions are don't leave the boundary, basically. Don't leave the domain, and they're free to do what they want. So they're continuous. They're not. So the Eulerian mesh is a discrete mesh, but then the fish actually moves continuously within it, and then it interpolates what it needs to.
Yep. You said that your fish can eat.
My fish can't.
Oh, okay.
Yeah. No. So I'm only looking at hydrodynamic cues, not environmental cues.
Yes.
I might have missed it, but are you assuming that the river flow is steady, so that a fish placed in a certain spot will always swim down the same pathway?
No, so there is stochasticity in the model, but the mesh or the CFD results are steady. They're required to be steady, yeah.
And then do the fish influence the flow?
No.
You had two fish with no feed each other.
They fish influence other fish, but they don't influence the flow.
So it's not through the flow, it's just through
Yeah.
the cues.
Yeah.
I'm confused. So your fish size is less than the mesh size, right? It's point-wise.
No, it's about a fish is about 4 cells long, about 4 CFD cells long.
So your one-way coupling assumption is not that valid in this case, is it?
How so?
Because there's a solid boundary in your flow which disturbs the fluid.
No, so if you sum up the actual amount of energy that a fish expends in a river, then that's of the order of magnitude of about 0.1 to 1 joules. If you compare that to the actual energy in the river itself, it's negligible. So you're right, two-way coupling would be more precise, but it's impractical because of the energy differences.
As we've got time, I'd just like to ask about the fish schooling idea. So obviously that's observed in fish schools. How might you incorporate that?
So I only look at predatory migrating fish, which generally aren't schooling fish. So, yeah. It could be included, definitely. It's just that they don't tend to care, to be honest. They don't. They're on a mission.
Yeah, thank you. I hope you can all hear me, and thank you for being still on the ball at this time. I'm talking to you today about evaporation from hydrogel in contact with water. This problem is motivated by something, well, I had hoped that there would be a window here to point out, but there's not. So we are looking at water ascent in trees. And I think all of you have learned in school, probably around year 8, 9, that water ascent in trees happens because you have transpiration in the leaves. Then I think everybody here has progressed and went to university, and there they learned, oh, but if we cannot pump water if we suck on the top higher than 10 meters, because we have to overcome a hydrostatic pressure of about 1 bar. So there is something to reconcile. The biologists, as one can read, for example, in this very recent review, they say, okay, the water in the tree is metastable, and therefore the water column doesn't break when the pressure becomes larger than approximately 1 bar, when we exceed the cavitation threshold. However, not everybody likes that idea. Particularly in dams, the idea is relatively unpopular, and most engineers have issues with the notion of water being metastable at extremely low pressures. So roughly every 20 years, somebody, like the last one was Zimmerman in 2004, attacks this view, which is called cohesion tension theory, and tries to find an argument why the cohesion tension theory is not correct. And the reason why I'm arguing quite defensive here is because the biologists are all convinced that it is correct. If this were a biology conference, I probably wouldn't dare giving this talk without a helmet. And also Zimmerman was actually quite violently attacked. 50 people wrote a letter to the editor of the journal and said, you cannot publish this. And the editor of the journal responded, well, we should publish it anyway. So even so, he himself didn't agree. So yeah, so this is one problem, if one thinks about this, is that the transport within the trunk of a tree is generally considered passive. So indeed, the really only source to move that amount of water is really evaporation at the top. So one problem is to find alternative mechanisms is what could it be? And I'm looking for one. And this is work in progress where I'm trying to build a very simplified tree in the lab, which other than the work of Wheeler and Strook, does not rely on very low pressures, but tries to actually overcome the hydrostatic pressure. But this is a bit of the future. But let's go to this. So my motivation is I try to simplify my tree. I have evaporation in the leaves through a hydrogel-like material called mesophile. And then we have the water-conducting tissue, the xylem, which in a conifer, this is one, a tree with needles, can be kind of sketched like this, where you have these cells which are called tracheids, and they're connected by little channels. And these channels are called bordered pits, where you have, again, a hydrogel-like membrane. And we try to rationalize this idea for the lab by building something which we call our lab tree, where we have evaporation from a hydrogel-like bead, which is our leaf. And then where we want to have evaporation, a membrane halfway on the way up the stem, if you want so, which is again like a bordered pit where we again have a bit of hydrogel. And the whole thing would be filled with water, connected to a reservoir, you have evaporation, and this should pull water up. As it is, if one does such experiments, because one cannot buy this, we are still manufacturing this. In fact, I think the day before yesterday, I got the first membrane ever. So I'm talking today about the single tree construction, which will not, in fact, will not resolve the big question I started with. However, it will give us some interesting idea about the behavior of hydrogels, and we will find, and I think I will be able to link this back towards the end. So what do? I have this, what I call a single tree. I have this bead holder. I fill this tube below the bead with water. This reservoir can move up and down. So one of my control parameters is the hydrostatic pressure just under the bead. The evaporation surface is encased in a chamber through which I cycle air with a defined relative humidity. And I measure the relative humidity of the air which goes in and which comes out, so it gives me a handle on the evaporation rates as difference. In fact, this is, by the way, a quite new setup, which only before I went on holiday started working. So the data I'm showing today is from my holiday, actually. Yes, it is quite challenging to keep this stable. Anyway, we have 4 of these beads in a cell, and on top we look with an SLR to measure the bead diameter. This is kind of the parameter we're essentially looking at, and what the camera sees looks like this. And this is not the bead. This is a hole in the bead holder, which is distorted by the curved surface of the bead. This is actually the bead in this picture. To give some idea of what is happening, this is a view on the bead. In fact, this is what happened over the holiday. After we put down the bead, we go away. I typically go away for 4 days, so I was at a wedding at this time. And let it equilibrate. This is shown here in this sample data. And then afterwards, we see here a response of the bead, which I evaluate, which is actually here from the fitting of which is, or the bead diameter is shown here by this blue line. And what you see is there's actually quite a lot going on. The bead seems to fall over. It seems to rapidly move around. If you play it, it's a bit slower. For what I don't have time, it may be even rotating. That's a bit speculating. So there's a lot of physics going on. But today I'm just talking about the evolution of the diameter. And as I said, the typical curve looks like this with the environmental data and with what I do to the reservoir below it. Note that the bead actually deforms. This is from a test experiment, which I probably will look at into the next iteration of the setup. I will present the data or look at the features of this big curve with all the, together with the result. I will compare them against the results from a simple hydrogel transport model. I really want to rush through this a bit because the basic idea was published by Bertrand 2016 for the drying of hydrogel beads. I reformulated this for a one-dimensional domain, which I put at the, by assuming that I have a semi-permeable membrane, put at the bottom in contact with a water reservoir at a given pressure. And on top, I assume I have evaporation, which I model by assuming that there's an equilibrium between the hydrogel surface and the air. That's something which so long I've thought about this talk, I'm not happy with that, but it was too late to change it. And you will see in a minute why. Note that this model can only be qualitative. There's no fitting at the moment. I use parameters which are sensible but are not necessarily related to our polymer beads. And as I said, all parameters are qualitative. All model results are purely qualitative so far. So if I change the relative humidity, and that's this black curve here in the relative humidity graph in the step function from 43 to 74%, when we see response of the beads that it immediately starts growing. Note, I had unfortunately some equipment failure here, and over the weekend nobody noticed. So that's why it probably doesn't plateau. It doesn't plateau off. Here is the flushing of the relative humidity chamber stop. However, as I decrease the relative humidity again, I see that the bead shrinks again. And what is quite interesting to see is that the evaporative flux becomes actually negative as I start increasing. As the bead size starts increasing, so the bead pulls water from the air, and at the same time, we see that the bead, we see that the evaporative flux goes up as we decrease relative humidity again, so the bead also evaporates in there. So the change really happens at the upper boundary. The functional shape is consistent with what the model predicts for a step change in relative humidity, but note that the order of magnitude is totally off. So this is something I'm working on. However, also the bead, actually the model predicts also evaporation from that the change begins at the surface. The other thing I did is I changed the pressure head under the bead. And again, we see a response. If we decrease the pressure head here, in this case by 20 centimeters, we see that the beads rapidly shrink. However, if we try to move the reservoir back up again, we see that we only have a small recovery if we really recover the original height of the reservoir, whilst for those cases where we only went back half of the height, we barely get any signal. This is not explained by the model at the moment. And also, again, the order of magnitude is really far off, which just predicts that we would recover. That we would not see any hysteresis. This could be either due to a more accessible configuration of space in 2 dimensions, or an experimental problem. Because if this is one of these speeds, when you take it off, you see that where it is sucked into the holder of the bead, into the bead holder, it forms a little dimple. So I'm almost done. So I would like to acknowledge support from the Leverhulme Trust, and we as a National Materials Innovation Center, and particularly our head technician who helped me carrying out these experiments whilst I wasn't around. And I think as a primary take-home message of this ongoing work is I think we have seen that if I control, or if I force the transport through a soft hydrogel, this leads to an internal pressure gradient. The model shows this clearly, it didn't show the graphs, but we, of course, need a pressure gradient, which leads to deformation. So now we could think about, if we return to our tree with this membrane halfway, what if we can have a deformed membrane? What would be the internal pressure gradient? And what would be the effect on the total hydrostatic pressure? Thank you.
think I'd like a little bit more clarification to
Yes.
explain this final thing you said about the single tree, the experiments you can do, the double tree are the ones you'd like to do.
I'm working, yes.
And how will that clinch it for the argument about the trees?
So I think the basic, so if we, so it may, so it is really, this is, as I said, this is slightly, this is the idea and our way of, the way how we plan this kind of leads to this idea. But when you deform the hydrogel, or when you deform the hydrogel, after, you would expect that the pressure gradient internally is, this leads to some pressure gradient because you deform the hydrogel, particularly if you deform it two-dimensionally, like the membrane sketched here, you would see there's a pressure gradient, or there's a concentration gradient at first, which then generates via the osmotic pressure a pressure gradient, and this would be directed probably upwards, so against gravity. That's the idea. But it's really a vague idea which I wasn't told not to put on the slide. So.